like to welcome everyone and let you know that I'm Angie Moxley. I work with Georgia College and State University in Milledgeville. I support this webinar technology for University of Georgia Extension and would like to introduce you to the host of this webinar session, Dr. Dan Suter. He's a professor of urban pest management in the Department of Entomology at the University of Georgia. He is located on the UGA Griffin campus. Dr. Suter has worked with the pest control industry since 1987. Dan would like to welcome you and introduce you to our speaker for today. Dan?
Well, thanks a bunch, Angie, and welcome everybody from all over the world. A lot of folks here in attendance from the southeastern U.S., but we do have people from all over the world. Dr. Mike Scharf is currently the O.W. Rollins Orkin Chaired Professor in the Department of Entomology at Purdue University in West Lafayette, Indiana. Dr. Scharf's research program focuses on basic physiology, toxicology, and applied aspects of urban pest biology, and mainly termites and cockroaches. And he teaches the course Insect Toxicology at Purdue. Dr. Scharf received his BS, MS, and PhD degrees from Purdue University in 1991, 1993, and 1997. After that, he was a postdoctoral researcher at the University of Nebraska and at Cornell University, and an assistant and associate professor at the University of Florida before joining the Purdue University in his current position. So welcome, Mike, and the floor is yours.
Okay, Dan, thank you very much. That was a very nice introduction. I really appreciate that. And it's really nice to be here today speaking to this audience around the world on this topic that's really one that's very interesting to me. As Dan mentioned, I actually teach insect toxicology, and so I spent a bit of time figuring out how to kind of present these topics, and so hopefully I can give you some really useful information here today that you can take back and apply in your day-to-day operations. So just starting off here, my overall goal is just this really broad goal, and that's just to improve the general knowledge of the audience, of you guys out there, of how insecticides actually work. And so, for example, sometimes you might have questions about, how does this product that I'm working with actually kill insects? Does it target the nervous system? And if it does, how does it do that? So I'm going to try to show you some of those things here today. Or, does the product work in another way, like an insect growth regulator, for example? Does it target insect growth and development, or the cuticle, or something like that? So knowing how insecticides work, how these tools work, is really essential knowledge for the industry. And so if you're kind of wondering, just how can that be? How is it essential knowledge? This next slide, I tried to just kind of rifle through some really key factors here that are reinforced to me all the time when I'm teaching toxicology and interacting with the industry on this topic. So, what do I need to know about insecticide classification and mode of action? Number one here at the top is safety. So, some insecticide modes of action have lower mammalian toxicity than they do pest toxicity, and some it's the reverse. So, knowing that about the products that we're working with can help us to use them more safely and in the right situations. Of course, the label dictates a lot of that too, but we can carry a little extra knowledge into that decision as well. Also, interpreting and the advertising and trade literature. I don't want to get too much into this, but sometimes the trade literature isn't quite technically accurate, and so you could know a little more than the industry in this case, and maybe make some better decisions about products. A big one that we hear a lot about today is pollinator health. We're trying to protect all those managed bees and native bees out there in the environment today. And so, for example, we hear a lot about nicotinoid insecticides causing problems, and one of the reasons that they they're on the radar screen, so to speak, is that they're water soluble and they can move in plants. And so we apply them to the soil and they can translocate in plants, and then the pollinators can be exposed. So that's the kind of chemistry knowledge that we can gain from insecticides here. Resistance management is really key today. Managing products to make them to prevent resistance from developing. And so knowing how insecticides work can help us use them in sequence better and make better decisions about how to rotate. Product sustainability. So each new product, insecticide product that reaches the market today costs hundreds of millions of dollars to get to the market. So there's a huge investment by the manufacturers there. And so if we can know how the products work and use them more wisely, we can make them more sustainable in the market. The next two, designing customized pesticide applications and situational pest management, those things, they can give you a competitive business advantage. For example, being able to design a customized pesticide application for a specific environment that can maybe create a new niche for you in your business. And situational pest management, not all insecticides are going to perform the same way in all environments. For example, in really wet environments or environments that are really greasy or oily, certain insecticides may not work as well. So that's another way we can look at this topic. And then finally here, customer communication and communicating competence. I mean, just knowing how to clearly explain how insecticides work to your customers, it sends the right message to the customers and the general public. So that's another reason to know a lot about how insecticides work and be able to apply that knowledge. Okay, so here's an outline of what I'm going to try to get through today. And I teach toxicology at the college level, and I know it's very difficult to cram all that information into 1 hour. And I'm trying not to do that today, but I'm just going to give you an overview of some very broad topics here. And first of all, a little background and introduction, just some basics of insect physiology. Looking at what's going on inside insects and understanding how those processes are disrupted. Just cover some basics of insecticide mode of action, in terms of neurotoxins and non-neurotoxins and those kinds of things. And then I'm going to go through the different neurotoxic insecticides and the non-neurotoxic insecticides that we have available on the market today, and try to give just a really broad overview of how those different insecticides work, and hopefully give you some good information there. And then try to integrate everything at the end and come back to looking at factors that affect how well insecticides work, tying it back to the chemistry and the modes of action and those kinds of things. So that's my task here today, and hopefully I'm going to do a good job on this, because this is a topic that I've really never covered before and with the industry at this level. So, and that's a good prelude to this message I want to give you, and that there are 2 additional sources of information out there. So everything I'm presenting in this talk, you can also go to. There was an article published in Pest Control Technology in October of 2011, which covers this material. And then also on the right here, Dan Suter and I published a little, what do we call it, a bulletin on insecticide basics for the pest management professional, which is available online and also at the University of Georgia at this web address shown here at the bottom. So even if you can't follow every specific detail of what I'm presenting here today, you have these other leading sources that you can go back to. And I'll bring these up again at the end if you can't quite get all the information at this time. Okay, so jumping into the first topic. So, this is insect physiology, a really brief overview. And so I think it's important to point out at the beginning that, everything boils down to chemistry. So it's the chemistry of the insecticides, You know, it's a chemical manufacturing industry produces these things. Everything is based on chemistry. But even from the side of the insect or inside of our bodies, you know, there's a whole lot of biological chemistry happening. And so everything boils down to that chemistry at a molecular level. But if we zoom back out, you know, we're talking about insect physiology that's being disrupted by the insecticides or affecting how the insecticides actually work. And so I wanted to start by just covering these critical components of insects. There are some things that are good to have a handle on before we dive into the topic of how the insecticides actually work. And so here I have just some really important features of insects. So if you take an etymology course, of course, you're going to learn all about these things right off the bat. And so I thought this was a good place to start as well. So here at the top we have, you know, this is a grasshopper, I guess. It's kind of sliced in half, and we can see, you know, here this blue structure going all the way through the ventral side, the bottom side of the insect. That's its nervous system. And so, you know, insects have a brain, and then they have all these ganglia, which are bundles of nerves at each segment for the most part. And then we have all these little peripheral nerves, these smaller blue lines that go through the whole body, and they control everything that's going on in terms of muscles and sensory apparatus, and there's information going away from the brain and traveling back to the brain, the nervous system, you know, through all these neurons. And so the nervous system is the most important target that we have for insecticides because it's controlling everything that's going on in the insect body for the most part. So that's the brain. And then if I could jump down here to the bottom right, so here's the muscles of the insect. So this is just this is not a real dissection. It's an illustration that was done for teaching purposes. But you can see that the insect body is just filled with muscles. And actually, one of our new insecticide classes, the diamides, which they actually inhibit muscle function, and they work very well, and they're very insect specific. I'll talk about those later. But our two big targets that we have, for the most part, are the nervous system and muscles of insects. And then we can think about other things that affect other factors that affect how insecticides work. And of course, we have the cuticle, which is, you know, on the outer surface of the insect. And the cuticle is actually made up of these living cells, the epidermal cells, but also there are non-living components which are secreted every time the insect goes through a molt. So that's the epicuticle, the exocuticle, and the endocuticle. And those things are all barriers to insecticide penetration. And so you know, it's a pretty formidable barrier that insects have that prevents insecticide from penetrating. And the cuticle is both on the outside surface of the insect, but also over here, if we look at the gut is made of cuticle as well. And so, you know, this is a termite gut, but it could be any insect gut with the mouth over here and the other end over here on the right, and everything in between. We have the foregut, the midgut, and the hindgut. And so if we have a bait insecticide, for example, or if a cockroach picks up some insecticide in its environment on its feet and it grooms that off with its mouth, it's going to enter. And so insecticides that enter the gut actually have to penetrate through to make it to the body and reach their target sites within the insect. And then over here on the right, this is just showing how complicated the tracheal system of insects is. And so insects, you know, they don't have lungs like we do. They don't have blood with hemoglobin in it, but they have this system of tubes called trachea that deliver air from the outside of the insect to the inside of the insect into specific cells, or clusters of cells and tissues. And so this tracheal system also can be a route of insecticide penetration. It's also important when the insecticide when the insect is actually poisoned, so it's breathing and acquiring air through this tracheal system that keeps it alive longer. And so, you know, this is just a really broad overview, but hopefully it gives you an appreciation for all the complicated processes that are going on within insects, but also how some really, really smart and creative people out there in the world and in industry and academia have discovered insecticides that can disrupt all these processes that I'm going to talk about. It almost seems like a miracle that we can have chemistries that can very specifically disrupt these processes. I'm fascinated by it almost every day in my line of work. So I wanted to move on then, after talking first about some things going on from the side of the insect, to coming over to the insecticide chemistry side and just talking about some very basics that are going on over on this side. And so with insecticide classification, just like we have organisms that can be classified, based on their morphology or their structures, like insects, mammals, whatever, birds, bacteria. Insecticides can be classified the same way, based on their chemical structures. And so over here on the right, I just have some chemical structures for different classes of insecticides, which, there won't be an exam on this. You don't have to know it here today. If you took my toxicology class, you might have to know these things. But somebody trained in the field can look at these different structures and actually put them into a class. And so we have, the cyclodienes up here, chloridane, here's a DDT, pyrethrins, organophosphates, pyrethroids, carbamates. It's very straightforward. And the reason I'm kind of belaboring this point is that these different chemical structures are what give the insecticides their, first of all, allows them to be placed into a class, but then that's what dictates what kind of target sites they interact with, which in turn impacts the mode of action of the insecticide. So these are all highly interrelated and interconnected concepts. Insecticide class, target site, and mode of action, they all go together in very predictable kinds of ways. So hopefully that's one of the things I'll be able to impress upon you for the rest of the talk, is just how very specific the insecticide tools that we have today can be in targeting very specific aspects of insect physiology. And on the flip side, at the same time, not impacting non-target organisms like mammals or people and pets, for example. We're in an area where we have very specific insecticides, and that's kind of exciting. So then drilling in a little more closely on target site and mode of action. So just to define what a target site is, so target sites are physiological locations within the insect. So target site it's a noun, as I described in my class, right? It's a place, within the insect. And so we can think of it over here. We think of the lock and key kind of analogy. A key has to be very specific to fit into the lock that it goes into, right? It has to be made by a locksmith, and it, the wrong key won't open the right lock, for example. So, when the insecticide goes into the target site, it meets up with it at a molecular level, and the key fits in the lock, and if it opens the lock, that's when we can get toxicity. So that's just a very general kind of analogy. Of course, so we can think of it as a lock and a key, but down here at the bottom here, I actually got this off the web. This is actually how our insecticides are designed today, for the most part, by people doing what's called computational biology and looking at chemical structures and figuring out how they fit into various proteins, which are the target sites. Again, this is beyond the the scope of what I want to have you understand today, but it's important to if we think of insecticides as these renewable resources, or I'm sorry, non-renewable resources, right? It takes hundreds of millions of dollars to get them into the market, because there's this very specific kind of research and development happening. And so, this is why we have such specific insecticides available to us today, through this kind of molecular docking kind of work that's going on. But anyways, bringing it back to the target sites. So the target site is a physiological location within the insect, And in general, we can have 4 modes of action or 4 kinds of action at disrupting these target sites. And so, you know, the insecticide can bind and stimulate a receptor, for example. It can bind and block that receptor, so it can just turn it off. It can bind, say, on the side of it and change its shape and modulate how it works. That's actually how pyrethroids work. They modulate the shape of sodium channels, which we'll talk about. And then we can have like enzyme inhibition. That's how our organophosphates and our carbamates work. And so these are just, you know, if we break it down, there are really only these 4 kinds of modes of action that insecticides can have. So, and I don't want to get any more detail than that because I know I'll probably hear about it if I go a little too far. But these are the kinds of things I think about a lot in my day-to-day research activities and also when I'm teaching the topic of insecticide toxicology to our students here at Purdue. So I should, you know, I would be remiss if I didn't talk a little bit about what LD50 is. And we can think about LD50, that's lethal dose that kills 50% of our test population. And that's how we determine how effective insecticides are against our target organisms and how effective they should not be against non-target organisms like mammals. And so, you know, LD50 is the standard measure of insecticide toxicity. And so really we can just say that the relationship between product toxicity and LD50 is inverse, which means that, you know, the higher the LD50, the lower the product toxicity. So products that are really toxic have a very small LD50 value. So I'm sure Dr. Kaler is going to talk about that a little in his next talk, so I'm not going to get into that too much. But we can just say that insecticides that are more toxic or more hazardous have lower LD50 values. It's really amazing that insecticides today are much more toxic to insects than to people or pets or other non-target organisms. Some are actually selectively toxic to the insect by over 10,000 times. So, and there's one example, the diamides like Rynaxypyr or chlorantraniliprole that we have available to us today. I'm not going to give any trade names, sorry about that, but they're very selective towards insects. And in fact, some of them the EPA said they don't need to have a signal word. They're so non-toxic or non-hazardous to mammals. But the manufacturers, to their credit, said, no, we're still going to give them a signal word. But that's just an example of how non-hazardous to non-target some of our insecticides are today. It's also important to kind of understand that because our insecticides are so active and specific, some of our newer ones, only very small amounts of insecticide that are placed in the environment actually need to reach their target sites to cause toxicity in the insect. You know, it's way less than 1 billionth of what's placed out there actually reaches the insect and its target site within its body. So that's just a little bit about LD50. Okay, so shifting gears here, I want to talk next about just our 2 very broad classifications of insecticides, and these are based on the target sites that they target. So we're talking about, I'm breaking these down by target site rather than insecticide chemistry. So even if you don't appreciate all the fine details that I talk about, you can at least, I hope you come away with these 2 very broad principles, and that, first of all, insecticides that target the nervous system, we have 5 major classifications there. So, you know, just try to appreciate that there are 5 different groupings of these things. And for insecticides that do not target the nervous system, so they're like insect growth regulators or respiratory inhibitors, or these kinds of things, we have four major classifications that I'm going to run through. So that's just a broad overview. And next, so I want to talk first about our insecticides that are neurotoxins, which maybe that doesn't sound like the best word or the best term we want to be using, but they are. They're insecticides that disrupt neurophysiology and disrupt the nervous system. So we can just say that the insect nervous system, it's made up of millions of nerve cells, right? So, which we can see here on the left. So neurons or nerve cells are cells just like any other cells in the body, except that they have this really nice ability to transmit electrical energy. And so you can think of them like electrical wires, basically, like we have in our houses. So that's the nerve cells. And then in the nervous system, we have, it's organized in different ways in different insects. And so, for example, in a cockroach, we have a very structured nervous system with one bundle for every segment. And in houseflies, for example, their nervous system's a bit reduced. It's kind of clustered into their brain and this big abdominal ganglion that they have here. And if you actually dissect them open, if you're really skilled, you can find these things, these structures very easily in the insect. So how does the nervous system actually work? So we can say that information travels through the nervous system in the form of electrical impulses. And then we have, here that's what this little electrical bolt, this little lightning bolt represents. We can have electrical energy that's actually moving down a neuron, a nerve cell, and then it'll come to these gaps. These gaps are called synapses, and we have neurotransmitters that cross those, and they bind receptors on the other side and meet up with another neuron, which then transmits that into electrical energy that moves down the neuron again. And so this process of electrical to chemical to electrical is happening as fast as the speed of light, if you can actually comprehend that. Electrical impulses move down neurons at the rate of microseconds, so millionths of seconds. It's really amazing to think about how the nervous system actually works. And it's based on this electrical to chemical to electrical signaling process. So you can imagine if you can disrupt that, that can lead to a negative effects in the insect, as we see with our insecticides. So, and how do we actually measure this? So, if you look at my little my icon, the picture of me as the speaker here, if you can see it, I'm sitting at this apparatus, and that's our neurophysiology rig that we have in our lab, and that's actually how we study the effects of insecticides on the nervous system. And so I don't want to get into the details too much of how that works, except that it's all tied to computers and electrical circuitry and being able to see the signals on a computer. But if we look down here in the bottom left, so this is a cockroach that's been dissected open, and you can actually see its ventral nerve cord there. And this is where we actually attach electrodes. It can measure things, as shown here on the left. So this is just a baseline recording. Those are the nerves just firing in the cockroach after it's been dissected open over time. And then if we apply a neurotoxin, in this case fipronil, you can see this really big increase in electrical activity, right? That's because we've caused a greater amount of activity in the neuron by applying the insecticide, and that can disrupt the insect. And so some of the things I'm going to be showing you in terms of knowing how the insecticides are affecting the nervous system, they're based on these kinds of neurophysiological recordings. And so there's been a lot of science done out there to actually establish the things that I'm going to be telling you here in the next few slides. I thought it would be really important to make that point. And so I'm going to cover very quickly here in the next few slides just these 5 classifications of neurotoxic target sites in the insect: sodium channels, chloride channels, acetylcholine receptors, acetylcholinesterase enzyme, and then we have combination products that target multiple things. I'll talk a little bit about those. And I'm going to skip the next 2 slides, but only say that this is how the illustrations would look in the articles that I referred to you at the beginning of the talk, and I'll bring back up at the end. just to give you some reference. So to make the point that if you can't follow everything I'm saying, you can at least go back and read about these in some more detail. The information is out there and readily accessible to you. So the first group I want to talk about are the sodium channel insecticides, and these are the pyrethroids, oxidizing bits, indoxacarb, and then the semicarbazones, which are mostly used for ectoparasite control. And we have metaflumazone there. And so what sodium channel insecticides do, if we look down here, sodium is a positively charged element or ion, and it moves into neurons as the electrical energy is flowing down them. It's the basis for the electrical energy that's actually moving along the neurons. And so these insecticides actually disrupt the sodium channel and either stimulate it and cause excitation in the insect, in the case of pyrethroids. Right, that's why we can look at a pyrethroid poisoned insect and see that it's shaking. It's because its nervous system is being stimulated. Whereas these other two groups actually block the sodium channel, and that leads to inhibition. And believe it or not, these things actually work the same way that the material that the dentist gives you to make you numb before he works on your teeth. These insecticides work in the exact same way. They block sodium channels. So that's sodium channels. The next group is chloride channel insecticides. And so in neurons, we have chloride channels that let chloride in. It has a negative charge, which means that it depresses the activity of the nervous system. In general, that's its job. It's kind of to keep everything in check, like a dimmer switch on your lights in your house to make them more dim. That's the same way chloride channels work in the nervous system. And so the big one here, we have the phenylpyrazoles, and that's fipronil, which is the only member of that class that we have on the market today, to my knowledge. Fipronil blocks chloride channels, so you take away that suppressive effect, and it causes excitation. Like I showed you in that nerve recording, we can apply fipronil to a nerve or to an insect and see, really elevated electrical activity happening. We also have the abamectins, which stimulate these chloride channels and cause inhibition in neurons. And so we have two very different kinds of action at the target site here for the chloride channels. Jumping ahead to, we have our acetylcholine receptor insecticides. These are exclusively, this is the nicotinoid class. So, this is a big group that we have available to us today. We have 6 products or so that are being produced in various forms out there. So what these things do, they work in a very different way. So we have normally electrical energy moving through one neuron, and when it gets to this chemical phase to cross the synapse, we have acetylcholine, which is that chemical neurotransmitter, and it'll bind the acetylcholine receptor in the next neuron, which sends the message along down the track there, so to speak. And so what the nicotinoids do is they bind and stimulate that acetylcholine receptor, which causes excessive excitation, downstream in the following neurons. And so that's how the nicotinoids actually work. They stimulate the acetylcholine receptor, and they cause excitation. Nicotinoids are very water soluble, and they move in plants, and they move in insects. That's what actually makes them a very good termiticide, because they can move between individuals with all that exchange that happens, for example, in social insects. And so, and this is how they work. They excite the nervous system. The fourth group are the acetylcholinesterase inhibitors. And so this is identical to, these are the organophosphates and the carbamates, which we don't really have available to us today in most places in the world. But for example, though, we still do have dichlorvos, which is registered for bed bugs, at least where I live, here in the U.S., and also carbamates, which still have some use in the turf and ornamental industry. And so these things are still out there. And so what they do is they inhibit the acetylcholinesterase enzyme. And so this is just like I talked about in the last the last slide. And I saw the question that just popped up about insecticide activation. I'll mention that here in a second. So we have acetylcholinesterase, which actually deactivates acetylcholine, right, that chemical messenger, and that has to happen on a very quick time scale for it to work. So these insecticides inhibit the acetylcholinesterase enzyme, and they actually cause excitation because we're not removing the signal anymore from the acetylcholine receptor. So a lot of the insecticides on this topic of insecticide activation, especially, for example, the organophosphates, they need to be in their raw form, in the form that they're produced or manufactured, they're relatively non-toxic. And once they make it into the insect body or into our bodies, they're converted into a form that is more toxic, and in many cases selectively toxic to the insect. That's not so true for the organophosphates, but with indoxacarb, which I talked about at the beginning here, the neurotoxins, it's a sodium channel blocker, and in the form it's manufactured, it's called a protoxin. It's not active at all, and then it has to be converted to an active form within our bodies in order to be toxic. And so we have this, it's called selective toxicity that's created through activation of the insecticide. There's a lot of complicated chemistry going on there, as you could imagine. But we can come back to that at the end as well. Okay, so I want to jump ahead here then to talking about combination products. So this is a big trend that we have in the industry today, and all of our combination products that we have involve nicotinoids plus pyrethroids. And currently, as I mentioned, I'm not going to mention any trade names here, but we have 4 products available to us, and they all start with T. So they're which is, I find to be pretty darn confusing, but they all start with T. And so when we're hitting 2 target sites at once, so we get this effect that's 1 + 1 = 3. So if, each insecticide by itself caused an effect equal to 1. If we add them together, we get this synergistic effect where it's 1 + 1 = 3. And this is called actually potentiation. This is a term that's actually scientifically based, but I think somebody out there trademarked it. But it means that you're affecting 2 different target sites at one time. And in this case, we have the nicotinoids, which are binding the acetylcholine receptor, and so they're kind of priming the neuron and turning it on. And then we have the pyrethroids, which affect the sodium channel and keep them from, basically just hold them open. So we have the on switch getting flipped on here by the nicotinoids, and then we have another factor that's just holding it open with the pyrethroids. And indeed, it's been scientifically proven that it is truly a 1 + 1 = 3 kind of effect. But also, if you get resistance to one of the insecticides, some people would tend to say that you have resistance to the whole product as a mixture. But again, the science isn't quite there to support that, but it's a point of debate, we'll say. So that was our combination products. And so remember, I mentioned there are 5 kinds of neurologically active products that we have out there, and I just went through those. Those affect sodium channels, chloride channels, acetylcholine transmission, acetylcholinesterase enzyme, and then our combination products here. So that's all I'm going to say about the neurotoxic insecticides, and then I want to come to these insecticide targets outside of the nervous system. And so these are our muscular calcium channels, insect growth regulators, inhibitors of energy production, and then finally our cuticle dehydrating dusts. So I'll try to hammer through these here. A big one, one of our really most exciting insecticides from the perspective of insect physiology and also selectivity to the pest is the diamide insecticides that we have. And so these work in a completely different way. We say that they're not neurotoxic, but they do affect the neuromuscular calcium channel. And so the way that muscles work is when calcium is present, the muscles contract. That's how our muscles actually work. And it's mediated by these calcium channels that when they're stimulated by the nervous system, they let calcium flow into the muscle, and that causes it to contract. And so what the diamides do is they stimulate the neuromuscular calcium channels, which causes the muscles to contract, which initially, in the initial phases, it looks like excitation. So the insect are in this state of, all their muscles are contracted and they're kind of tremoring while they're exposed there. And then over time, as they burn up their energy, they just kind of show inhibition. So they're very, very flaccid, and they're just not moving and lethargic. And so these are the insecticides that I mentioned that they actually had no signal words were required by the EPA, meaning that they were so selective towards insects and so non-hazardous to mammals that the EPA said they did not need a signal word, but they still do have a caution signal word, which is that lowest category. So really interesting insecticides that we have here in this group. We have the insect growth regulators. Quickly, I just want to kind of move through these. You know, insects, they have these outer exoskeletons, and they undergo metamorphosis of different types depending on the insect groups that we have. You know, insect development it's really intricate. And so we have these very detailed hormone cycles that are happening as the insect moves from its egg through its larval or nymphal stages to adulthood. You know, and the big one is juvenile hormone. So every time the insect goes through a stage, it has a pulse of juvenile hormone, which keeps it in the immature state. And then once they molt to an adult, a lot of things have to come together in order for the cuticle to actually form properly. And that's really, you know, really key for the insect to be normal and function properly and reproduce and be mobile and all those other factors. You know, it's a really intricate process with the hormones and the cuticle. And so we have our IGRs, which insect growth regulators. We have two classes, the juvenile hormone analogs and the chitin synthesis inhibitors. And what the juvenile hormone analogs do is they artificially raise the juvenile hormone level even when it's not supposed to be present. And so that leads to cuticle deformation and extrajuvenile stages. So here's an adult cockroach that looks like a nymph, and it's got wings that are all malformed and curled up. You know, this is a classic symptom of insect growth regulator juvenoid exposure. And then we have the chitin synthesis inhibitors, which inhibit the enzyme that produces chitin, and that leads to, you know, cuticle deformation and death during molting. And here, what you can see down at the bottom, some really nice pictures of termites that are affected by chitin synthesis inhibitors. They show this hunchback kind of pattern, you know, even after they molt, which means they are not very happy termites. So that's the IGRs. The next group are the inhibitors of energy production, and we have basically a lot of different modes of action here, but are different chemistries, different classes. Hydromethylnon, chlorphenapyr, we have our fumigants like sulfurylfluoride, and then we have the wood preservation agents and other materials like the DSOB, disodium octaborate tetrahydrate, and boric acid. So all of these things just disrupts mitochondrial processes in the insect. And mitochondria, they're the energy power plants of insect cells, or of all cells, no matter what they are, plants, microbes, mammals, you know, people, insects. We all have mitochondria in our cells, and that's where we make all of our energy, and we also respire. That's where the nuts and bolts of breathing actually take place, down there in the mitochondria. And so I don't want to get into specifics, but all of these insecticides inhibit respiration in mitochondria, which symptom is paralysis that you can see. And this is because the insect isn't making any energy, and also even nerve cells have mitochondria in them. So some of the first effects we can see from these insecticides are just on the mobility of the insect. They just don't move very well when they're poisoned by these things. And so a lot of these as well, coming back to the idea of activation, these materials, especially chlorphenapyr, need to be, they're produced in a relatively non-toxic form, and then once they make it inside the insect's body, they're converted to a very effective inhibitor of energy production by disrupting mitochondria. Okay, and then the fourth category here is the cuticle dehydrating dusts. And so this is both silica gel and diatomaceous earth. And where these things come from is they're based on silicon, which is like glass, right? It's a glassy-like material. It's a very powder. It comes in nature from diatoms, so fossilized exoskeletons of diatom organisms, invertebrate organisms. So they crush those up, you get this glassy powder. And what it does is, if we look over here at the cockroach cuticle, you can see this nice waxy sheen on the outside of it. That's its waterproofing layer. That protects it from dehydration, basically. And so these materials will abrade that outer layer or absorb the waxes that allow the insecticide to lose its water. And when you're really small, like an insect, just losing a little bit of water can have really profound impacts on your physiology, your ability to maintain your body. So that's how the dehydrating dusts work. So even though they're not really directly toxic to mammals, you definitely do not want to breathe these things, because if they make it in your lungs, they'll be there for the rest of your life, and so you want to have proper breathing protection when you're using them. So they're not completely inert from our perspective. Okay, so in the remaining 5 minutes, I just wanted to talk about a few factors that kind of feed upon the chemistry ideas that I've been talking about, and then bring it back to more practical kinds of knowledge that you can apply in day-to-day operations. So the first thing to keep in mind with insecticides is stability and persistence. So most insecticides are very oily in nature, which that's how they work. It helps them to cross the cuticle and membranes and make it into the insect and reach their target sites. And so, target sites are usually in membranes, which are really oily environments. And so, like you can see here, oil and water don't necessarily mix. And so that creates a lot of problems, which are solved by formulations, which I'll show in the next slide. We don't apply insecticides in their pure raw form, because they'll degrade by UV light, like with pyrethrins and pyrethroids. Without being formulated, they will degrade very quickly in UV light. Some things are also very water soluble, and they'll just go away when it rains, for example. And so that's where formulations come into play. And so I'm not going to really talk about formulations, because I'm sure Phil Kaler is going to get into this, I hope, in the next talk. But we'll just say that formulations are complex mixtures of the active ingredient, of inert ingredients, food attractants in the case of baits, and stabilizers. So formulations are really complex mixtures of which only a small proportion is the actual active ingredient. And so, these things enhance stability. Formulations enhance stability and extend longevity. They enhance safety for the applier. They make the product easier to handle and mix. For example, getting that oily insecticide to dissolve in water. That's the formulation. The formulation helps to make that happen. Insects have behaviors that make a lot of our insecticides today work. A lot of these insecticides are very water soluble, and so that can allow them to move from one insect to another, like with cockroach secondary and tertiary kill after they feed on baits. For example, also flea larvae are exposed to insecticides through the feces of adults. So here we have some flea eggs and larvae, and so the adult is defecating out insecticide off the host pet that they're feeding on, and that's how the larvae are exposed. And a lot of that is made possible by the ability of the insecticide and its chemistry to dissolve at just the right amount and move in the feces of the insect from adult to offspring. And then social insects practice trophallaxis and allogrooming, which I'm sure Bob Vandermeer is going to talk about in the next time this webinar meets. But trophallaxis and allogrooming really enabled insecticides to move through social insect colonies very well. That's why we have things like fipronil, Termidor, oops, I named the product there, but fipronil and other soil insecticides that are non-repellent that can move from one insect to the other very well. Also sanitation, it's really critical to make our insecticides work properly. Of course, excess food in the environment competes with baits. Clutter creates excess harborage that can't be treated, giving the insects refuge from the insecticide. And this one's especially a big factor. Dirt and grease tie up the insecticides just because of their very nature. Remember, they're very oily. The insecticide molecules themselves are more oil than they are water. And so they'll absorb onto dirt and grease, etc., and that can affect the way they work. And so this just underscores, how IPM and sanitation are still important for making our chemistries that we have available work. And then finally, the last factor I wanted to mention here is resistance. Right? Knowing how insecticides work is really key to resistance management. So, perhaps the #1 cause of callbacks of cockroach accounts is resistance, and we know also that bedbug resistance is widespread to pyrethroids. Maybe not other materials, but definitely things that target the sodium channel, like pyrethroids. And so product rotation is key to sustainable long-term success in these two pests. That's the message we're pushing, and we're sticking to it. Even mixture products we feel need to be rotated. And so to effectively rotate, as shown here on the right, you need to be able to know which products you're using and to be able to rotate to different products accordingly. And so I'll just, I think I just heard the chime go off and I've reached my time. So just the big summary point is increasing your insecticide knowledge. It can just make you a better pest manager. And that can have all kinds of impacts on sustainability of pest management in the industry, preventing resistance, communicating a really positive message, positive image to the customer, and just keeping the industry operating at a very high confident level, and giving a good image for the whole industry. That's really why it's useful to understand this topic. And I'll just close here and say that there are 2 additional sources of information out there. There's this article in PCT from October 2011, and then the Insecticide Basics bulletin published by Dan Suter and myself, which we feel are really good resources to go to for this. And so I will close there, and hopefully I've kept us on time, and hand it back over to Dan.
Thanks. Thank you, Mike. I think everybody joins me in thanking you for that just outstanding presentation. We did have a couple questions come in. One of the questions that had come in earlier during the registration process was about rotation, but you did address that towards the end there, and you did, for many of the folks online, probably saw your earlier session a couple months ago on insecticide resistance, and you explained that quite well then. But one of the other questions we had come in, you can see it down there in the chat box, this comes to us from India. Are cockroaches resistant to delta mether? I know you see some pyrethroid resistance in German cockroaches, but he's talking about 2.5% delta mether.
Mm-hmm. Right. Well, so that 2.5%, that's a very high concentration, so we would think that there should be some efficacy there. But, I've seen it many times. Resistance can really happen to anything that's overused. And so just, upping the rate or the concentration of the insecticide can get around resistance for a while. But if you don't move to a different product, it's going to you're going to lose that insecticide for a very long time in that population, maybe permanently. So yeah, resistance to pyrethroids was in roaches was common 25 years ago, and it's still there today. So.
Yeah, there aren't many other questions coming in, Mike, but I had one on, you had talked earlier about differential hazard between mammalian toxicity and arthropod toxicity. Could you talk a little bit about what, maybe over the past 10, 15 years, what manufacturers are doing to, I don't know if design is a good word, but look at actives that show less mammalian toxicity and greater arthropod toxicity? Is there any truth to that?
Oh, absolutely. Yeah. So I mean, that's driving our manufacturing industry today. So, virtually all the discovery that's happening is in that direction of finding new active ingredients that are very specific to arthropods and have a low mammalian toxicity. So, but you can imagine all the secrecy that goes around that, of course. But a lot of that comes from the molecular modeling, like I showed, which is, I can't even wrap my brain around it. It's very heavy on the chemistry and the computer science, but yeah, a lot of our new materials that we're going to see soon and in the future, in the distant future, will have been designed by this knowledge of pest specificity, and that comes from all the genome sequencing that's happening today. You can look at the different target sites and know how they're different.
We had one other question come in here just a second ago, Mike. It says labels and safety data sheets really don't always state the class or family of insecticide. Is there another source that this information could be found?
Yes, I would say so. The Insecticide Resistance Action Committee, or IRAC, maintains a website. So I'm sure you could. It's irac.org, IRAC.org, I believe. If not, you can Google that. They have a very nice listing of all the different insecticide groupings, and they update them at least once a year, maybe twice a year or more. And it is extremely helpful for designing rotation programs, but it's also a good resource for how the insecticides are placed into classes.
Mike, it's a fantastic job. I'm getting lots of good comments coming in, so really do appreciate the time that it took for you to put this presentation together. I know you had a lot to work on, but we really do appreciate this. Thank you.
Yeah. Thank you, Dan. My pleasure.
Yep. Thank you.